Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in creating a better tomorrow for millions of people out there. Today, we have the honor of being joined by Dr. Luba Perry. She is the co-founder and chief executive officer of Reconstruct Bio, which is a really innovative venture emerging from Harvard's Wyss Institute, aimed at redefining the fields of medical reconstruction and aesthetics, with initial applications of their really groundbreaking technology on breast reconstruction and augmentation. And they have a multidisciplinary team of experts. They have been developing their technology BioImplant, which is a living bioengineered tissue. It's created from a patient's own cells to ultimately provide safer, more natural alternatives to the current standards, which are associated with many significant drawbacks and health concerns. Dr. Perry also serves as a senior scientist at the Wyss Institute for Biologically Inspired Engineering, working on the 3D organ engineering initiative back in 2018, and is leading Wyss Validation Project, aiming to fabricate vascularized functional tissues for transplantation. And her interest in tissue and organ engineering focuses on vascularization implantation studies using these complex surgical models. Her background is in molecular biology, pharmacology, and biomedical engineering. Did her bachelor's in biology, her master's in molecular pharmacology, and her PhD in biotech, all at Technion, Israel Institute of Technology. And she also has industry expertise from a vascular gene therapy company, now known as VESSL Therapeutics. A lot of really cool topics to get into today. Honored to have with us Dr. Luba Perry. Thank you so much for taking the time to come on the show.
Hi, thank you so much for having me. I'm excited to be here.
Yeah, I'm excited to have you. I'm looking forward to this topic. A lot of really interesting themes that all sort of come together at a really interesting application or several applications for humans. Would love to start off, Luba, by just handing things over to you for a little bit to talk a little bit more about you. I noticed sort of before you got into everything organ engineering and organogenesis. A lot of your early work had to do everything from mitochondrial proteins that regulated apoptosis to certain glycoproteins that regulated scar formation, all really interesting parts of sort of the wound healing regeneration equation. What got you interested in the space to begin with? You've been on a really interesting journey.
Yes, definitely. So I started with biology at the Technion back in Israel. And then kind of was very, very interested in all neuroscience. And I thought I really liked pharmacology and definitely was drawn for like translational, like more applicative science. So I went and did my master's at the molecular pharmacology department at the medical school at the Technion. And I realized that initially I thought that's what I wanted to do, but then I realized I really liked the science. I was not very passionate about what I was doing. And it was still a lot of like basic research, more than I thought, while still pharmacology, but really understanding receptors, understanding like processes. And I'm like, oh, this is not exactly what I want to do. And I knew I wanted to do a PhD, but I knew it's not that. So I took a few years off and I went and worked in the industry, which really, I think, changed everything for me. And because I was very, very lucky to end up where I did, and that was a vascular gene therapy company. At the time, it was called MGVS, so multi-gene vascular systems. And it was funded by a cardiologist from the Carmel Hospital in Haifa. And I was working in the R&D department. I was doing all our QC for our large animal studies, where we were developing these biosynthetic grafts for dialysis. So that was amazing. That was the first time I kind of discovered the field and the space of regenerative medicine, and specifically vascular tissue engineering. And I just fell in love immediately. I'm like, oh my God, this is incredible. So I kind of, okay, this is what I wanted to do. So I decided to go back after 2 years in the company. I went back to the Technion, but this time to the biomedical engineering department, and worked in the lab of Professor Shulamit Levenberg, who's really one of the world's experts in vascular tissue engineering, and was working specifically on fabricating vascularized skeletal muscle for transplantation. So a lot of animal work, a lot of really doing exactly what I wanted to do, of building these vascularized tissues in the lab and then implanting them into animals and see how this engineered tissue can actually integrate and connect to the blood vessels of the patients. Eventually the patients, initially the animals, and that was incredible feeling. So I knew that I was doing exactly what I wanted to do. And after that, we moved to Boston, so around 6 years ago. Initially, I did my postdoc at the Wyss Institute. So I joined, it was a newly established 3D organ engineering initiative at the Wyss, which is co-led by core faculty members Jennifer Lewis and Christopher Chen. And also we have Sangeeta Bhatia as an associate faculty at the initiative. So I worked initially on like different projects, mostly on all still vascularization of engineered tissues. So I was mostly working on liver. And then around 4 years ago, we had this idea of applying for internal funding at the Wyss Institute that is really geared towards commercialization. So on top of course, we have very cool science, very amazing technology that we develop and publish in high impact journals, but the Wyss Institute is not just that. We actually want to bring technologies out to the patients. And then there's a lot of work on top of the technical development, of course, thinking about the business, actually, if you want to bring it out there, and thinking about like your patients and your end users and your payers and like the entire and your investors, like the entire story. So that's how like Reconstruct was really born 4 years ago when we applied for this funding, and then we got it. The project was named Engineered Vascularized Tissues for Therapeutics. So it was very broad. We had a very cool bioprinting technology that could fabricate these small-scale vascularized tissues, but it was all in vitro. And we wanted to see, can we scale that up? Can we build like a bigger tissue with that? And can we implant it into the animals? So initially, we decided to start with fat tissue, basically as a technical proof of concept tissue. While being the 3D organ engineering initiative, all of our other projects are actually liver or kidney or heart, really organs. But here we had so much to do on the technical side to really build a bigger fragment or tissue or organ. We decided to start with fat because technically speaking, it's easier to fabricate a vascularized fat tissue than like a vascularized heart or liver, because on top of just keeping the tissue alive, it also has a very complex functionality. So we decided to do the fat just as a proof of concept, but then we were planning to like move on to our organ of choice. And then when we had that funding and we started thinking about more than just the technical development, and talking with KOLs and talking with surgeons, I noticed that the plastic surgeons were so excited about the possibility of having a vascularized fat, and that this is something that they could really use in the clinic for their reconstructive patients. And also they all started talking about breast reconstruction. And then, so maybe I'm going like really into reconstruct. So I don't know if that's a part of your question or not.
Really is, and I'd like, before we get too far into sort of the business side of it, I wanted to go a couple places and sort of in parallel, because a lot of the work that you do and you did leading up to this has its proxies. For instance, if you were just looking at your dissertation, where you were looking at sort of the microenvironment in this particular case, effective cell composition on integration of transplanted vascularized engineered muscle tissue, where you were looking at, okay, we have this muscle, we need the proper endothelial cells, and you discovered some really interesting stuff during your dissertation, that adult endothelial cells worked a lot better than umbilical ones, and so forth. When we're looking at breast tissue, again, as you were mentioning, okay, fat, right? But there's other stuff there in terms of fibrous tissue and glandules, and I don't want to speak out of turn. What are some of the things that you learned sort of early on in terms of sort of what is the proper cellular composition to a breast, in the sense that the heart is made up of certain cells, the kidney, so forth, that you have to think about to create that proper vascularization, because don't want just fat, right? So talk a little bit about sort of what went into that thought process, and again, it was paralleling with your dissertation there, but I think it relates kind of nicely.
Yeah, so actually one of the amazing things specifically with so not necessarily breast, but just soft tissue reconstruction is that we have a really good source. Like the cell sourcing is not a huge issue as it is for organs, where it becomes a really big challenge where you're taking the cells from. You most likely you will be taking sick cells if you are taking them from a diseased organ. So you don't want that. And then you have to go somewhere else, like iPSC, stem cells, and then it becomes like a huge complex thing on the cost, because you have to start with cells that you need to expand and differentiate. But then also practically talking, just getting those cells. So when we are thinking about the soft tissue, what is good that we can just use the patient's own fat, like people when they're doing liposuction, right? And in that fat, you have your fat cells, the adipocytes, but then you have a lot of other cells, like endothelial cells, that are already present in the fat, like mesenchymal stem cells that are present in the fat. There are pre-adipocytes, there are like other cell sources as well, but these are probably the most important ones for us, are just the fat cells, and then the endothelial cells for the blood vessels, and their supporting cells. Because really in tissue engineering, we are trying to find out, when we're building those organs and tissues in the lab, we really want to take the minimal amount of cells needed to build that organ, right? And then we are relying on regeneration. So you would have other cells either differentiating to the cells that you need, or just coming from the environment and populating that area. So really for the fat, as I mentioned, for us, is just having the adipocytes and the pre-adipocytes, but then for the vasculature, having the endogenous adipose-derived microvascular endothelial cells that are present in the fat that we could separately isolate, expand, and then seed them in our bioprinted vessel-like structures, basically, to line them with endothelial cells.
So no, that's awesome. And in addition to that work, you also spent a lot of time, obviously looking at what I guess, a proper scaffold is. I know you worked with different types of scaffolds in the past when you worked on muscle cells and the hemodialysis work, and also sort of the spectrum of cytokines and other growth factors that are involved in the process of organogenesis and these processes of regeneration normally. Talk a little bit about what the sort of the scaffold is in this particular case, and some of the things that you're learning about the cytokine, the paracrine milieu in the breast tissue that you really have to sort of focus on for this process.
Yeah, so basically with the scaffolds and all of my work during my PhD, and then also like my earlier postdoc project on the vascularized liver, we were relying on the endothelial cells' capacity to self-assemble into blood vessels, into small blood vessels that are called the capillaries, basically. And that process, we could really use scaffolds that support that culture of the endothelial cells with the supporting cells under the right condition to self-assemble into this capillary structure. But then, and what is new with Reconstruct is realizing that we need to build, that we want to scale that up, right? And build a much bigger tissue. We cannot just scale up that type of scaffolds, either synthetic or natural, with a lot of very complex, beautiful microvascular structures, because they're just not, it's not enough. And if you look at an organ, it really has a lot of degrees of vascularization, starting from these capillaries, but then you branch into larger vessels and to really even larger veins and arteries, right? So this is something that we wanted to mimic when we build a larger tissue. And then those scaffolds don't really work anymore because you are not able to fabricate these hierarchical vascular structures within. So what we are currently doing, what we're doing with Reconstruct, is basically, we still have those capillaries and the small vessels that are self-assembled from the endogenous endothelial cells. But then we have a larger bioprinted vasculature, and you cannot really print in scaffold, the type of scaffold that I was using for my PhD, like collagen scaffolds or like PLLA, PLGA scaffolds. You are not able to print with them. So what we are basically doing, we are printing within a very dense cellular matrix of these, what we call them organ building blocks, which are basically clusters, well, not basically, but we can refer to them as like clusters of cells, right? And we are using more like hydrogel as our matrix to combine everything. So one example could be fibrin, where you mix fibrinogen and thrombin to cross-link and really connect all these cells and the aggregates. Could be aggregates, could be organoids, could be spheroids of cells together. But before you cross-link that, you actually bioprint the sacrificial material within those cells that you can evacuate. So we don't really have like a scaffold, like a porous scaffold that you would imagine. It's really mostly your cells, your own cells, because we really want this to be like a native tissue.
That's great. One other area I wanted to ask about based on your previous work. I noticed you'd also, alongside a lot of the work that you were doing in vascularization, you had an interesting paper from a couple of years ago, again, working on muscle, talking about innervation. And, this is something that, I spent a teeny bit of time in sort of the regenerative biology world and, sort of the importance of sort of healthy peripheral nerves in regenerative processes has sort of been documented for something like 100 some odd years now. But I'm just wondering what you learn, not as much in terms of obviously creating these natural implants, but in sort of the what's the word, the intercalation, when you obviously have to surgically implant and sort of integrate this new tissue with the rest of the system. What is the importance? What are you learning about sort of the importance of the nervous system here in sort of reconnecting this new tissue with the body?
Yeah, well, definitely super important, especially when we're talking about something like the breast tissue, right? I actually came back last week from the annual meeting of the American Society of Plastic Surgeons, and they had a session exactly on that, how there are so many new technology, like techniques now in the clinic to preserve sensation and to really do innovation for both implant-based reconstruction, but then for autologous reconstruction, where you transfer the tissue. Because currently, in breast reconstruction specifically, when you are doing the autologous one, meaning you don't want the artificial implant, so you cut your own tissue from a different part of your own body, like the lower abdomen, for example, and the plastic surgeon only cuts an artery and a vein. So when you move that flap, what's called a flap, to the area of the chest, they're only anastomosing one artery, one or more veins, basically. But they are not connecting like lymphatic vessels. They are not connecting nerves, at least not as a standard practice. But now I was so happy to hear how there's more and more surgeons realizing how important it is to actually innervate the flap as well. So this is hugely, hugely important. And there's definitely, like with a good engineered tissue, you can also see like small vessels from nerves, sorry, from the area that are connecting, that are invading your engineered tissue. And this is something that I've seen like during my PhD work and with like these vascularized scaffolds, but that is a very, very slow process, right? And takes a long, long time. And so ideally, maybe Gen, I don't know, 4 of our bioimplant, we would have the blood vessels, we would have a lymphatic system, and we would also have nerves that you can immediately connect all of them. But this is, I think that the most important thing is the vasculature at this point, and we can definitely use some of the techniques that are currently being used in the clinic for breast reconstruction to just use veins, either from a different part from the patient or engineered, not veins, sorry, nerves.
The material, for Reconstruct Bio, obviously the core platform or I guess focus is going to be on breast reconstruction. But you mentioned that it clearly has applications when we just look at therapeutic and sort of aesthetic across a variety of potential cosmetic surgeries. And you also mentioned in the materials things about metabolic disorders, and I was kind of interested in that as well. If you could sort of take us down both those paths of where you see sort of beyond breast reconstruction, because I know obviously there's a lot of cosmetic surgeries that you could potentially use this technology for, but also, we hear about sort of fat and sort of the therapeutic applications of white fat versus brown fat and all that. And I'm speaking out of turn here, but what are some of if you could take us down a little both of those corridors in terms of other aesthetic applications and some of the sort of the other sort of whether metabolic disorders or other therapeutic disorders you potentially see applications of technology for?
Yeah, so basically, initially we see ourselves starting in the reconstructive space, so reconstructive breast reconstruction, but then, of course, any type of soft tissue reconstruction, whether if it's congenital defect or from trauma, or if you remove a tumor, there are other incidents where your only option today is actually to harvest that tissue from a different part of your own body. So this is, yeah, you solved one problem, but then you created another problem in your donor site. So this is definitely not ideal. And with all of the reconstructive, we're talking about the regular standard, like white fat, the fat that you couldn't just take from the liposuction. And so basically we're like doing liposuction, we're still using your own tissue, but it's just minimally invasive procedure to get your fat and then form it into this engineered flap that is vascularized with a vascular pedicle that you can connect in the area of interest. So this is for reconstructive. Then for cosmetic, of course, if we're still staying in the breast surgery space, breast augmentation, the #1 most performed plastic surgery worldwide. There are over 2 million women that are doing breast augmentation every year, even though the only option they have is the artificial implants, right? Like the silicone implants, which like as you mentioned initially in the introduction, there are so many things that are wrong with these implants, but this is the only thing, and women will want to have bigger breasts, just how that's how it is, and that's their only choice with all the limitations and all the problems that come with it, from like tiny things to like, oh, you get lymphoma from your breast implant. So it's really a huge variety of complications that can happen. But then any other augmentation that you could think of, like your butt or like in your face or like anything, basically, because it's really customized, in size, in shape. So as long as you need something like a fat tissue to either fill a void or to make something bigger, we can do it. So all of this, we are talking about the standard white fat, the regular fat that you can just take from the liposuction. But then talking about the brown fat, with currently there is a lot of research, so it's all very early, all preclinical, most of it small animals, not even large animals. Of all these research that shows that brown fat, which is or beige or brown fat, which are metabolically active versus the white fat, could really, if you either turn your white fat into brown fat, or you inject yourself with brown fat, it really can help reduce weight. It can help with glucose tolerance, and really with like my studies, they shown that it can help with obesity and like type 2 diabetes. So there's a lot of like research of how we can use that brown fat to treat metabolic disorders. And the biggest problem with brown fat, even if you like transform your fat to brown or inject yourself with brown fat, that because it's not vascularized, there's a lack of vascularization, it either dies or it turns back to white. So like one of the biggest challenges in all of this like brown fat thing to treat these metabolic disorders is actually vascularization. And this is exactly what we have solved. That we can turn this white fat into brown. And there's a lot of protocols out there, and kind of pretty standard, that you can do ex vivo browning of white fat. So for us, we are thinking to do the same cell sourcing, so do the same liposuction, but then you can turn that fat into brown in the lab, vascularize that, and implant that to the patient to actually keep the brown fat brown and to increase the metabolism of the patient. And this would like surgery, right? But still one surgery that would last for a lifetime, and not like drugs that you have to take like every day. And if you stop taking them, you regain all the fat back. But this is like very early on for us. We're very focused on the reconstructive cosmetics, so like the white fat, but this is definitely one application, an additional application that we can pursue.
No, I think what you're talking about with the topics of sort of cellular reprogramming, dedifferentiation, transdifferentiating to different tissues. I think that again, that it fits very nicely into sort of what's happening in this whole domain in 2024. So, and I'm excited to hear about those opportunities. One other thing, you took us here. I just have to ask. I apologize for this question because you were mentioning size and shape and things like that. I mean, in sort of the world of organogenesis research, some of these areas like size and shape when it comes to a heart or a kidney or a part of a spinal cord or whatever, these are kind of still mysteries in many ways, right? About sort of what says that a heart is a certain size, doesn't, I say it can't be the size of a watermelon or a walnut. That won't be acceptable. When it comes to breasts, obviously, they come in all different shapes and sizes. And I'm just like, how is that? I apologize for this question, but how is that dictated? Obviously, with silicone implants, right? Or someone wants a triple E or something, fine. That's not a living product. But in this case, if someone comes to you and wants something much larger than sort of the original genetic blueprint dictated, does that cause a problem? I just, it popped in my mind when you were mentioning the other.
That's a great question. And technically, if we are expanding the cells and differentiating them, then we could potentially reach any size we want, but that would be very expensive. Right now, we're going into very extensive cell culture that is like similar to like the cell therapy space, where you really reach like a very, very expensive price for your product. So to make it really cost effective, we are not going to be able to make huge bioimplants. It would really depend. It will be patient specific, depends how much fat we can take from the liposuction. Even very low BMI patients, below 18, have enough for like a small implant size. So we can definitely generate small and higher. It won't be like crazy amount unless someone really wants something very big, and then it's super customized for that patient, and it will be more expensive than the standard.
When it comes, I mean, obviously one sees the extreme potential on the therapeutic side, on the aesthetic side to what you're doing at the company, but clearly there's also some sort of lower hanging fruit here in terms of screening models. In terms, I mean, I can assume that a sort of having a three-dimensional vascularized breasts. In the lab may be extremely useful in testing new drugs for breast cancer. At the same time, I know there's been work out there that been focused on sort of the biomechanics of the breast during for sports injuries and potentially sort of the biodynamics of what happens in tumor formation over time. What are some of your sort of the thoughts on sort of using what you're developing there for some of these lower hanging fruit opportunities in drug development, drug discovery, and so forth?
That's an interesting question. Been thinking a lot about it because for me, like we might have to do it eventually to generate some revenue before. But for me, I really want to build this tissue for transplantation. There's no question about it. And I don't want to like waste time on doing other things. And I think for me, talking with patients that have these artificial implants or had the complications or had the surgery, made it clear that we need a better solution for those patients and we need it yesterday. Like this is not acceptable that currently there are now definitely no good solutions. That there is a solution, and that's why a lot of people are not really innovating in this space. If you really look at like those artificial implants that have been used since the 60s, they have not changed much. And women are not happy, and women are suffering from these complications. So I really want to get to patients as fast as we can and to have it for complete reconstruction following mastectomy. So I don't want to get kind of, what is the word, distracted with other applications. While important, but there are many others. There's like organ chips. There are like so many other modeling companies that are not aiming and will not be able to build a tissue for transplantation, so they can focus on that. I think this is our mission, and for me, this is like top one priority.
No, I like it. Go fast, go big, go home. I get it. I get it too. No, it's totally. But that being said, what should we be on the lookout in terms of Reconstruct Bio as we get close to 2025? Any other major public-facing things happening in terms of conferences, both scientific or investor-focused? Anything else that I missed that's coming up as we get towards the new year? Please take the floor.
Yeah, so in 2 weeks, I think in 2 weeks, yeah, I'm going to attend the AdvaMed, the MedTech conference in Toronto. So we'll be a part of a panel there as a part of the MedTech Innovator Plastic Surgery track that Reconstruct got accepted to. So we're one of only 5 companies that got into that track, which is like less than 5% acceptance rate. So that is coming up. And then in general, we are fundraising. And we were very lucky and fortunate to get internal funding from the Wyss Institute for the last 4 years to really bring us from like in vitro proof of concept from like a mouse cell line that we differentiated into fat cells to where we are today, where we are able to fabricate a human tissue, small scale, but to implant it into animals, to keep it alive, to keep it perfused. So we really achieved all of the milestones that we were planning to achieve within academia. And now fundraising to officially spin out from the Wyss and conduct the large animal studies.
Excellent. Well, I'm rooting you on. I love great startup stories like this, and you clearly have the right scientific background. You're in the right place. So again, best wishes for you and your team as you further develop these technologies and bring them out to the public. Again, for everybody that's going to be listening to this particular episode of the show across the podcast network, so who'll be watching on the YouTube channel. Again, you've been spending time with Dr. Luba Perry, co-founder and chief executive officer of Reconstruct Bio, senior scientist at the Wyss Institute for Biologically Inspired Engineering. Luba, I want to thank you for taking the time out of your schedule to talk to us for a little while about everything you're up to. Obviously, thank you for what you do for millions of women out there. And as we like to say on our show, thanks for helping to create a better tomorrow via the science and technology that you championed. A really awesome story.
Thank you so much for having me here. It was
It
really
was
great.
great having you.